and you don't remember, or you want to refresh your memory on some of these concepts, at least you can go to the appropriate chapter and look at your notes for that chapter. Okay. Number one, for chapter one, define and distinguish between anatomy and physiology. Well, you know that anatomy is all the physical features of the human body, its structure. And then physiology is the functional aspect of that. So by definition, you should know that anatomy is structure, physiology is the function that is possible because of that. Two was identify the levels of organization. We started off with the chemical level of organization. We went to cell, tissue, organ systems, and organism. And we had a whole chapter on chemical, we had a whole chapter on cell, we had a whole chapter on tissues, which make up organs, and then we started looking at a few organ systems. I think we actually studied 4 different organ systems this semester. So we had to get a whole bunch of background information before we could do that. Now, number 3, it says identifying osteocytes and neurons as what level of organization. Well, osteocytes are part of what system? Bone, right? Bone, okay. But those are actually cellular, aren't they? That would be the bone cells. Okay, what is a neuron? A neuron is a cell of which system? Nervous system. So both of these are representative of cellular level of organization, right? But these are different kinds of cells because they have different kinds of jobs. Number 4, sarcomere. If you'll remember, what is a sarcomere and what level of organization does that represent? What system is it associated with? Muscle, but it's not a muscle, is it? Is it a muscle cell?
A tissue.
It's not a tissue, it's not a cell, it's just a functional unit. It's made up of chemicals. That's all it is. It's chemical level. Okay? A sarcomere is just one of those little contractile units that's made up of what 2 proteins? Actin and myosin. Those are chemicals, aren't they? All right, you should be able to give a definition for metabolism and homeostasis. They're related, but what is metabolism?
The chemical reaction in your body.
It's the sum of all the chemical reactions that are necessary to maintain homeostasis. But what is homeostasis by definition? It's that relatively constant internal environment, right? And all of our organ systems have to work together to maintain homeostasis. Yes?
Now, you said the sarcomere is a part of the chemical system, right?
A sarcomere is part of the muscular system, but it represents chemical level of organization.
Okay.
Does that make sense? It's not a cell, it's a part of a cell. Okay, now we identified 11 major organ systems in chapter 1. We only covered 4. I think we did the integumentary system, we did the skeletal system, we did the muscular system, and we did the nervous system. So that means you're going to be covering 7 additional systems in the second semester. You're going to start with endocrine, and then go to cardiovascular, digestive, urinary, so on and so forth. There are 11 major organ systems in the body. We covered 4 of them. All right, now all of these organ systems are regulated by a feedback system. It could either be negative feedback or positive feedback. Which type of feedback is the predominant way we maintain homeostasis?
Negative feedback.
Negative feedback. So if you have any deviation from a normal parameter, you're going to have steps that reduce that deviation. There are only a couple of instances where we have positive feedback that's important. What are those? Do you remember? Childbirth, and there was another one mentioned. If you are bleeding profusely, the response would be an exhalation, clotting. The clotting is going to be accelerated. That's going to be, that's going to be a positive until actually the bleeding stops. Okay. So, number 8, and number 9 are really important in that this is all the nomenclature that we've learned that we need to use for anatomy. You should know the regional terms, like the different regions of the body, like this was brachial, this was cubital, this is antebrachial, palmar. You need to know all those regional terms, body region terms. You also need to know all the directional terms, like superior, inferior, medial, lateral, those kinds of terms. Proximal and distal. Where do we use proximal and distal? Arms and legs only. Okay. We also learned all the different planes of the human body. What is the plane that cuts you right down here and gives you right and left? What is it this way? Frontal or coronal. What if it's across? Transverse. What if it's at an angle? Oblique. Okay. And that gives you different parts of the body. Okay. What if it's right in the center this way? Mid, sagittal. Could it be over here and still be sagittal? Yes. Okay, sagittal just gives you right and left parts. Okay. Then we had a number of body cavities and some serous membranes that were associated with them. So, did you have a cranial cavity? Did you have a vertebral cavity? Do you have a final review study sheet?
Yes, ma'am.
Okay. We are down to number 9. Okay. So you had, these were part of the dorsal cavity, right? Cranial and the vertebral. Okay, the anterior cavities made up the thoracic, abdominal, pelvic. What separates the thoracic and abdominal? Okay, is there anything separating the abdominal and the pelvic? No. Did we have some some serous membranes that were associated with these different cavities? We had a cavity specifically for the heart, didn't we? What was it called?
Oh,
Pericardial. Did we have a cavity for our lungs?
yeah.
Pleural cavities. Did we have cavity in the abdominal pelvic? Okay. You have a question? Here. Okay. So we had serous membranes associated with those. If you'll remember, the membrane on the organ was called the visceral, right? And the one that was outside of that defined the space, was the parietal. And we named them based on the particular organ, the particular cavity. What's between the two membranes? Serous fluid. And those membranes produce it, don't they? Okay. We are at number 10, and this is the beginning of chapter 2. Okay? Chapter 2 was all the chemistry that we had to learn. It was a long, long chapter, and we only have 5 questions here. It was a long, long, long chapter. 3 was 2. You should know what elements are by definition. Basic substances that make up the material world. Atoms are the smallest unit of an element. Do you know any of the subatomic particles of an atom?
Protons,
Protons, neutrons, and electrons, and you know where they are and what their charges are, right? What are ions?
charge.
Atoms with a charge, or groups of atoms with a charge. Do you remember the types of bonding that we had? We had ionic, which was between metals and nonmetals. We had covalent, which was between nonmetals. And then we had hydrogen bonding. Do you remember that? We talked about hydrogen bonding particularly in water or between other substances that are dissolved in water.
What was the first one you said?
Ionic bonding, ionic, covalent. You should remember the characteristics of each of those. Ions, you have, ionic bonding, you've got ions formed, right? It's the opposite charges. Covalent is where you have sharing of pairs of electrons. And hydrogen bonding is between hydrogens that have a partial positive charge and another atom that has a partial negative. We saw that illustrated in water. You should know the difference between acids and bases. Do those influence pH? Sure. Okay, so acids are acidic because they release hydrogen ions. So hydrogen ions are an important ion that is related to pH. What are buffers? What do buffers do? Buffers resist pH changes, so they try to keep the pH fairly constant. Okay, now you should know that sodium and potassium ions are important in the body. They are electrolytes. In other words, those ions are important in that they can carry an electrical current. Okay, electrolytes. Do we have electrolytes in our tissue fluids and in our blood? Yes, you do. Is it important for muscle function? Important for nerve function? Important for pH? Yes. So all of the electrolytes are charged atoms that have very important functions. We know that sodium and potassium are involved in that synapse, that neuromuscular junction. Okay. Now, number 12. We looked at two important groups of molecules. One group are called inorganic, and one group that we studied more extensively were the organic molecules. What makes a molecule organic? What does it have to have?
Carbon.
And hydrogen. Carbon and hydrogen. Got to have carbon and hydrogen. If it does not have carbon and hydrogen, it's not organic. Inorganic molecules are usually formed between metals and nonmetals. Okay, metals and nonmetals, and that's usually ionic. Covalent molecules are generally the organic molecules. Okay, 13. We talked about 4 major groups of macromolecules. You should remember that carbohydrates, lipids, and proteins are associated with foods. What are nucleic acids? What are the 2 nucleic acids?
Yeah.
And
Alright.
RNA. DNA and RNA, they're made up of nucleotides. You should know the monomers for all of these. Which of these first 3 do we primarily use for energy? Carbohydrates. What do we use next? Lipids. We don't generally use proteins to generate energy. You can as a last resort. Proteins are often structural. Proteins can be enzymes. We've got proteins do all kinds of jobs in our bodies, but they're not the type of molecule that we generate energy from. It's usually carbohydrates. Nucleic acids, DNA and RNA. Where do we find DNA in the cell?
Nucleus?
In the nucleus only. It's always in the nucleus unless the cell is undergoing division. Where do we find RNA? In both, actually. It's made, the RNA is assembled in the nucleus, and then it leaves to go to the ribosome to make proteins, right? So RNA is primarily involved in protein synthesis. DNA is involved in heredity and in regulating all the cellular activities. Enzymes. Enzymes are proteins. What do they do?
Catalyze?
They do, but how do they affect the rates?
Speed it up.
They speed it up or increase it, okay? Chapter 3, starting number 15, Chapter 3, identify cellular structures and their functions. We spent a good bit of time in lab on this, but we spent even more time in lectures. So you should be able to identify all the cellular structures. You should be able to tell me something about their function, okay? So go back through those lists of structures and find out their function. What's the role of the mitochondria?
Oh, electricity.
It generates energy, right, in the form of ATP. What does a lysosome do?
Break down.
It's digestive, right? It has digestive enzymes. What is the primary function of rough ER?
Protein.
Makes proteins because it's rough, because it has
Ribosomes.
ribosomes. Okay, what about smooth ER?
Lipids.
Primarily lipids, yes. Okay, so look at all those structures and come up with their primary function. Now, our cells are compartmentalized using a lot of membrane. Now, the cell membrane or the plasma membrane is made up primarily of lipids and a number of other proteins. So, what is the primary lipid that we find in our plasma membranes? Primary lipid that makes up the plasma membranes. It makes a double layer, makes a bilayer.
Phospholipids.
Phospholipids. Another lipid that's in our cell membranes is cholesterol. Cholesterol is found in all animal cell plasma membranes. It makes the membrane a little bit more rigid and reduces permeability. Do you remember that we had a lot of proteins in the membrane?
Mm-hmm.
And they had specific functions, didn't they? Some of them were involved in transport, some were not. We had some enzymes, some were receptors. We have a variety of proteins that are found in cell membranes. So you might want to remember that some of them were called integral, some were called peripheral. Now, number 17, we looked at the membrane proteins and we know that 2 of them are involved in transport. Those were the channel and the carrier proteins. Now, there's 2 different types of transport, active and passive. What does active tell you?
You need ATP.
You have to have energy, right? Active transport requires what kind of energy? ATP. If it's passive, what does that mean? No energy required. What is the only thing that makes substances move across the membrane if it's passive?
Concentration.
Concentration gradient. Okay. Y'all remember all that word? Okay. That phrase, rather. Okay. Now, another thing that we looked at was tonicity, and this was talking about movement of water across the cell membrane. We did an experiment where we exposed our red blood cells to different solutions that had varying amounts of sodium chloride. Okay? So tonicity is talking about different solutions that have varying concentrations of a solid. What were the 3 types of solutions that were possible?
Hypotonic, isotonic, hypertonic.
Yes, okay. And those tell you something about the concentration, right? Now, we use sodium chloride solutions. What was isotonic? Do you remember what the normal was? It was 0.9. 0.9. So if I gave you a question with a concentration, would you be able to tell what kind of tonicity it was? Okay. Well, that influenced movement of water. So did their shapes change?
Mm-hmm.
Okay, did one of them cause the cell to rupture? Which one did that? Hypo. Which one caused it to shrivel up? Hyper.
What were they called again?
What now?
What were they called? One was crenation and
Crenation is the shriveling up.
And-
Hemolysis was the rupturing of the red blood cell
what's
due
the
to a
concentration?
hypotonic solution.
What were the concentrations for the hypertonic and hypertonic?
Hypertonic is anything over 0.9. Hypo is going to be anything less than 0.9. Does that make sense? Okay. Okay. 19, talking about DNA, talking about 3 different events that occur with DNA. What does DNA replication mean? If you replicate DNA, what are you doing?
Making another copy.
You're making another copy. Why would a cell need to do this? Huh? So it can divide, right? It's got to make copies before it divides. Okay, so we're going to talk about that whole process in just a minute. But you need to recognize what these words mean. DNA replication is where you make identical copies of the DNA for cell division. These other 2 are actually associated with protein synthesis. Is the information for proteins on the DNA? Yes. And we can transcribe that information into an RNA strand, can't we? So we can transcribe the DNA information into an RNA code. And that RNA strand carries the information for making the protein. Where does that RNA strand go?
Messenger.
To the ribosomes. It goes to the ribosomes, right? And once that RNA strand gets to the ribosomes, is that information translated? And translation means the RNA strand is read and a protein is made from that information at the ribosome. Is this sounding familiar?
Wait,
Transcription is where you take DNA and change it to RNA. Translation is where you take RNA and you change it to a protein. Does that make sense? You're using that RNA information, which came from DNA, to make the protein. That simplified? Okay, DNA to RNA, and then RNA to a protein. Okay, number 20. The cell cycle, by definition, is the time period when the cell is carrying on its normal functions, but is it getting ready to divide too? So, what are the stages of the cell cycle? Do you remember?
G1,
That's cell division.
G1, S, and G2.
That's interphase, and then the last stage is M, right? So G1, S, and G2 is interphase. It's just when it's carrying on its normal function, and then M is when it actually divides. So there's M refers to mitosis and cell division, but that's a totally different process. If you're looking at the cell cycle, you have 4 stages, G1, S, G2, and M. So do you know what's happening in G1? Do you know what's happening in S? Do you know what's happening in G2? And then M is cell division and cytokinesis. Okay? So, do you know the stages of cell division? Yes, and that's when all those chromosomes are getting distributed so we can get 2 new cells, right? Okay? So you should know what's happening with the chromosomes during prophase, metaphase, anaphase, telophase, and at the end you have cytokinesis.